Hey Mustangs, in this video we're going to be taking a look at autosomal single gene disorders, so let's go ahead and start. So in order to understand gene disorders, we have to understand heredity. So everything we've learned about heredity so far is going to help us see how gene disorders are going to be passed along. So in the concept of heredity, we have parents that pass on their DNA to their offspring, and their DNA contains instructions for the traits and characteristics of how their offspring are going to look, how they're going to be put together, how they might even behave. So here in this image here, we see two red-headed people. They're going to have red-headed children. Every single time they pass on their DNA, in this case, that's the only possibility, unless there's some sort of mutation that may occur that would cause a child to be born with hair other than the color of red. Now, this has nothing to do, like I said, with gene disorders, but it's the idea of heredity, that the child's traits are determined by the DNA that's passed along to the parents. So the DNA instructions that are in the parents' gametes, the sperm or egg, is what determine the overall traits of the child. Now here's a picture of actual family that has children with a gene disorder here. So you can see the two sons to the right over here clearly look a little bit different than everybody else, and it's because they have a disorder, and we'll be talking about this specific disorder at the end of the notes here. So before we go any further, make sure that you go to the class website and print out the handout in order to fill in these notes. If you want, you can take the notes by hand, but I figured it'd just be easier if you go to the class website and print out the handout that's already there. So it should be listed as gene disorder notes or autosomal single gene disorder notes from last class. So just go to the class website, print those out. Now this first one here, you actually don't have to write anything down. So with the basics, one thing that you need to understand before we go into the gene disorders is that humans have 46 chromosomes in every diploid cell of their body. So every cell that's not a gamete, that's not a sperm or egg, is going to have 46 chromosomes. Okay? So it doesn't matter if it's a nerve cell, a muscle cell, blood cells, white blood cells, pancreas cells, it doesn't matter. They all have 46 chromosomes in it if it's a diploid cell. Of those 46 chromosomes, there are 2 sex chromosomes, and remember this is humans here, so 2 sex chromosomes. The sex chromosomes are the chromosomes that have DNA instructions that help the person develop into a male or female. Now remember, when we're talking about a chromosome, so we often draw our chromosomes like this. So if that's a chromosome, there's usually homologous pairs. Remember that if you were to zoom in on a chromosome, it's basically just DNA, a DNA double helix wrapped around histone proteins. Okay? So out of all the 46 chromosomes that humans have, 2 of them are sex chromosomes that have DNA instructions for turning a person into a male or female. They also have some other instructions as well. So it's not just like the DNA in a sex chromosome is only connected to the determination of sex, but there's other instructions in there as well. Now there are 44 autosomes. So the autosomes are the chromosomes that basically contain all the other information for all the other traits. And then a karyotype is a picture of all 46 human chromosomes, all lined up together from the largest pair, the longest pair of chromosomes, down to the smallest pair, and they're all in homologous pairs. So here is an example of a karyotype. So you can see here the longest pair all the way down to the shortest, and then the sex chromosomes are always put last. Okay, so sex chromosomes will always be last when you're looking at a karyotype. Okay, now looking at the first 22 pairs here, 22 times 2 is 44. So these are the first 44 chromosomes. Those are called the autosomes, and then you have the sex chromosomes last. Now, one way to remember the difference is that if it's not a sex chromosome, then it's automatically an autosome. That's the easiest way to remember this. So here's our sex chromosomes here. Everything else, therefore, is an autosome. So everything else in this top section, all of these are going to be your autosomes. Okay, so autosomes, pairs 1 through 22, 22 times 2, the first 44 chromosomes, and then the last 2. So 44 plus 2 equals 46 altogether. All right, nothing to write down on this one. I just wanted to kind of lay it out on the different types of genetic disorders that there are out there. So a genetic disorder is a disease or disabling condition that is a result of mutations in the DNA. So any disease or disorder that is caused by messed up DNA in some way is going to be referred to as a genetic disorder. So there's two main types of genetic disorders. So if we take genetic disorders as a whole, diseases or disabling conditions based on messed up DNA, there's gene mutations where genes get messed up and they can lead to the disorder, and then there's chromosomal mutations, which we'll talk about next time. So an autosomal gene disorder, which is the title of these notes here, single gene autosomal disorders, these are disorders that occur in the genes of pairs 1 through 22. So once again, taking a look here, here is pairs 1 through 22. So these are the first 44 autosomes, or chromosomes, and those are the autosomes. So if there's a mutation in the DNA here and here, or even just one of them, one or both of them, that could lead to a disorder, some sort of condition that makes it hard for a person to function. It could be extremely debilitating to where it leads to death, or can cause problems for the person throughout their entire life. So anytime you have a gene disorder in pairs 1 through 22, the first 44 chromosomes, the autosomes, that is called an autosomal gene disorder. Okay? We'll be talking about in class as well, and in the next notes about disorders that occur in the sex chromosomes. Those are usually called sex-linked gene disorders versus the autosomal gene disorders. Okay? So what is a gene disorder? A gene disorder is when the instructions in 1 out of 20,000 genes get messed up. And so we have about 20,000. Scientists have been going back and forth on this. They haven't really said exactly a number, so approximately 20,000 genes. So out of all this DNA, remember this is all DNA here, DNA wrapped around histone proteins. Throughout all this DNA, there's about 20,000 different genes. If someone has a single gene disorder, that means somewhere within these genes, there might be a space here. Either one or both of the same gene gets messed up. That leads to a disorder. That leads to a condition that makes it difficult for the person to live. How do these disorders occur? Well, within that gene section, if there's an insertion, a deletion, a substitution of one or more bases, that's going to mess up the DNA instructions. If the DNA instructions are messed up, then the amino acids won't be put together correctly. And as you know, if the amino acids aren't put together correctly, then the protein is the wrong shape and the protein can't do its job. So what this really comes down to is when the DNA instructions are messed up, the proteins are made incorrectly, and something in the body isn't functioning correctly as a result of that, because every protein needs to be made correctly in order to do its job. And without that protein being made correctly, that person has a problem. So go ahead and fill in this here. So you have this on your paper. Go ahead and fill in for both the normal and the mutated, and then come back to the video. So when you fill it all in and you use your chart and everything, you see that the instructions here, and this is a short little sequence of DNA. This is the normal sequence of what you should get. These, of course, down here are the amino acids that connect together. Over here on the other side with the mutated, there's one single letter that's mutated. So this is a substitution in this case. So it's not an insertion, it's not a deletion, it's a substitution. That substitution changes the DNA instructions, and instead of putting together serine, alanine, and lysine, you put serine, threonine, and lysine. And this is going to change the shape of the protein, and whatever that protein was supposed to do, it can no longer do its job correctly.